All right, this is the super fast run through of the B1 and B2 content for the new IB biology curriculum. Let's jump in. Carbon forms the backbone of all organic molecules. Those are the molecules that make up living things. The reason that it's so important is because it has 6 protons, usually 6 neutrons, and 6 electrons. Because on the innermost electron shell, it's going to be stable with 2 electrons. Carbon has 2 electrons there. Then on the next electron shell and each one after it, it's stable with 8 electrons. But since carbon has 6 total, there's going to be 4 electrons in the outer shell. And in order to get to 8, it's going to want to make 4 bonds, which is the maximum number of possible. So since carbon wants to make 4 different bonds, it's going to form that structural backbone of every organic molecule. Okay, here's a simple methane where carbon is making 4 bonds with hydrogens. So say, for example, those blue electrons are those 4 on the outer valence electron shell of carbon. Hydrogen just has 1 electron, indicated here in red. And so now, since they are sharing these electrons through covalent bonding, that carbon is happy, it's got all 8. Hydrogen's happy because it's got these 2 on its innermost shell where it's stable. And this forms a nice, stable methane molecule. Okay, we usually would write that molecule like this two-dimensionally, but you should know that it is a tetrahedral shape. Okay, we're going to talk about carbohydrates. Carbohydrates are made up of sugars. These are usually ending with the suffix, ose, like glucose, which is a monosaccharide, a single sugar. Maltose being a disaccharide, which is just 2 glucoses bonded together through a condensation reaction. Okay, just a reminder that you should be familiar with the metric system, also known as the International System of Units. Okay, a monomer is an individual subunit. Polymer are multiple. Dimer isn't listed there. It's going to be on a future slide. That's going to be 2 specifically. So glucose plus glucose joined through a condensation reaction, which produces water, is going to make a disaccharide, generally a dimer. Okay, the bond that's formed between 2 carbohydrates is called a glycosidic bond. So right here, we have a hydrogen being removed from this hydroxyl group. We have this OH group being removed, which is going to allow this carbon, okay, to bond to this oxygen. That CO bond, okay, is our glycosidic bond, and we've produced maltose. Okay, so here again, monomer, dimer, polymer, those are going to be general terms for any of our types of biomolecules. For sugars specifically, saccharide refers to those sugars. Okay, examples of monomers are glucose, fructose, dimers, maltose, like we just saw, or sucrose, which is a glucose and a fructose. Polymers include glycogen, starch, and cellulose, which we'll be talking about in a second. Okay, so remember this is a reversible reaction. Condensation reactions can take 2 glucoses and make a maltose. Okay, but we can also do hydrolysis, which is lysing or splitting by addition of water, okay, which can break that disaccharide back into 2 monosaccharides. Okay, plants store their excess carbohydrates as starch, and we have 2 forms of starch, amylose and amylopectin. Okay, amylose is made up of all carbon-1, carbon-4 bonds. Remember our carbon numbering system, we're going to start with an oxygen. The carbons then go in order clockwise. So this is carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, and carbon 6. So therefore, if this is carbon 1, this is carbon 4 of the next glucose, those 2 carbons are going to bond together in a condensation reaction. Okay, and we're going to make amylose, which is just this long chain of carbon 1 to carbon 4 bonds. It's like, just think of a bunch of people holding hands, carbon 1 to carbon 4. All of their sort of hands are joined together. Now, amylopectin is another form of starch, which has mostly those same exact 1,4 bonds, but occasional carbon 1, carbon 6 bonds. And those are the locations where we see branching. So mostly 1,4 bonds, occasional 1,6 bonds at the branches. Okay, this is just going to show you again a hydrolysis reaction a little bit more closely. Okay, so monosaccharides, we're going to see hexose sugars where they have 6 carbons. Okay, glucose is the one you need to be familiar with. Okay, ribose sugars are going to have 5 carbons, so are deoxyribose. Some properties of glucose that make it really great for our energy input. It's very soluble, so it's easy to dissolve in blood plasma and therefore be able to travel throughout the whole body. Okay, easily transported in blood, chemically stable. Okay, and we're able to get energy from it when we oxidize it through cellular respiration. There are two types of glucose. You should be able to differentiate between alpha and beta glucose. Sometimes you'll see this written as alpha D glucose or beta D glucose. Don't let that confuse you. Okay, so I like to think of beta glucose as the one where we follow the pattern, where we have this carbon going up, so then this hydroxyl group's down, up, down, up. It just continues that up-down pattern. So the OH on beta glucose is going up, and the opposite is the case on alpha glucose, where it goes down, up, down, down. Okay, so those alpha glucoses are the ones that we are going to find on those starches we just mentioned, amylose and amylopectin. Those alpha glucoses are going to make those nice chains. Amylose does become a helical shape, okay, as does amylopectin, other than those 1,6 branches. Now, glycogen is how excess sugars are stored in animals. It has a similar structure to amylopectin like we see here, but there's even more 1,6 bonds, so we have additional branching, even more than amylopectin. Okay, so what we can do is we can store these carbohydrates, and then as those sugars are needed, right, we can do hydrolysis and break them off one by one in order to be able to use those glucoses in cellular respiration. Okay, by storing glucose as starch or glycogen, it helps it be more compact, okay, relatively insoluble, and fairly easy to add to and take away from. So this is going to be our medium-term storage of energy, as opposed to glucose itself, which is our short-term. Now let's compare that to cellulose. Cellulose is made up of chains of glucose, but these are beta glucose. Okay, beta glucose, remember, follows the pattern where the OH group goes up on carbon 1. So what that means is when they form these 1,4 glycosidic bonds, they are every other one is inverted. So it's up, down, up, down, up, down of the entire glucose molecule. What that does is those alternating molecules, okay, that's going to allow for hydrogen bonding to occur between these regularly spaced hydroxyl groups. So because it's beta glucose and we have that inverted up, down, okay, we're not just going to have these chains, but we're also going to form, okay, these big structures. These are going to make up cell walls. This is cellulose, and it's incredibly strong. It's like a sheet because of these additional bonds that are forming. And so we can't digest cellulose for this reason. It's got that high tensile strength, strong covalent bonds, okay, making up plant cell walls. Okay, quick note on glycoproteins. These are just going to have an oligosaccharide chain. Oligo means few, so just a few sugars bound to a protein that's going to be embedded in a cell membrane, and they're used for cell recognition. A good example of that is going to be our red blood cell types. The reason that we have different blood types, are because of our different glycoproteins that are listed on each of our red blood cells, depending on how we inherited those blood types from our parents. Okay, shifting now then from carbohydrates, let's talk about lipids. Lipids are another group of biomolecules. They're nonpolar, they're hydrophobic. They include oils, fats, waxes, and steroids. Oils are going to have that low melting point. So they're going to be solid only at very low temperatures. At room temperature, oils are going to be liquid. Fats, on the other hand, are solid at room temperature, but liquid at body temperature. That's going to be an example like butter, where we know butter is solid, but as soon as we start to heat up butter, maybe we're spreading it on some toast or throwing it on a frying pan, it's going to melt. Okay, waxes, though, have a much higher melting point, right? We know that in a candle, for example, that's not going to melt until it gets really hot. Okay, and then these are fourth class, which are steroids. Steroids have these 4 rings. Okay, and one thing that you need to be able to do is recognize the difference between a carbohydrate and a lipid. And one major difference is with a carbohydrate, we're generally going to have a 1 to 2 to 1 ratio of carbons to hydrogens to oxygens. Let me go back and show you what I mean. Okay, so not a good option right there. Here we go. Let's look. Okay, so here we can see that we have 6 carbons, 6 oxygens, and 12 hydrogens, okay, in this glucose molecule. That is our 1 to 2 to 1 ratio. So we have the same number of carbons and oxygens. And so if you see that, then you know that's going to be a carbohydrate. So going back to our lipid, okay, we're going to see that there's way fewer oxygens compared to carbons. Remember that all of these angles are each carbons, carbon, 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 carbon. Okay, whereas here we only have 1, 2, 3, 4, 5 oxygens. So we have way more carbons than oxygens, and that is how you're going to be able to tell that it's a lipid. Okay, our classic lipid is going to be a triglyceride made up of a glycerol and 3 fatty acids. Those fatty acids then go through those 3 condensation reactions to make our triglyceride. Again, notice how few oxygens there are compared to the number of carbons. This is a lipid. Now, the difference between a phospholipid, which makes up cell membranes, and a triglyceride is just that instead of that 3rd fatty acid tail you find on a triglyceride, right, we have our phosphate in our phospholipid. This also gives it that amphipathic property of having one side that's hydrophilic. You can see those little charges there. Okay, and then we have our nonpolar hydrocarbon tail, which is going to align in our membrane with the tails towards the inside, away from the water. Okay, a fatty acid has a carboxyl group at one end, okay, and then a methyl group at the other end, which is just a CH3. Okay, the length of the chain is variable. It's usually between 14 and 20 carbons. Now, fatty acids can either be saturated or unsaturated. I like to think of saturated as saturated with hydrogens. You can see here that everywhere there could possibly be a hydrogen, one exists. On the other hand, an unsaturated fatty acid has at least one double bond. Now, because carbons always want to make 4 bonds, okay, if there's a double bond here, this carbon is making 4 bonds without having another hydrogen bonded. And so unsaturated fatty acids have at least one double bond, and they're therefore not going to be saturated with hydrogens, so to speak. They're not going to have hydrogens everywhere. Now, we have a couple different types of unsaturated fatty acids that you need to be able to distinguish between. Okay, first up, if they just have one double bond between the carbons in the hydrocarbon chain, that's monounsaturated. Okay, if they have more than one, then that's going to be polyunsaturated. Also, notice how these unsaturated fatty acids have a bend in them. Let's distinguish between cis and trans fatty acids. Now, in cis fatty acids, which are natural, we're going to see this bend form because these 2 hydrogens are missing, because the 2 hydrogens present are on the same cis side. Now, this is going to be really healthy because it's a bent fatty acid, so it's not going to pack together as tightly, right? We don't want those fats to form those plaques, which could lead to diseases of the blood system, okay? In contrast, trans fatty acids, where the hydrogen is on either side, these are generally going to be produced artificially, and they cause that fatty acid to be straight, which means those fatty acids can pack together way, way tighter. I like to think of these as like spaghetti, for example. If you think of a box of uncooked spaghetti, you can fit like hundreds of spaghettis in one box, like so packed really, really tightly. Versus picture if those spaghettis had a bend, right? You might be able to get 4 or 5 in there, right? Instead of hundreds. And so this is going to just prevent that tight packing. It's going to be much healthier to have those cis fatty acids. And that's going to be nearly all the unsaturated fatty acids found in nature, usually liquid at room temperature versus trans fatty acids are solid at room temperature. Okay, the good news though is trans fatty acids have been banned because of concerns by the FDA for the health concerns associated with them. Okay, so you should be able to identify if you're shown a fatty acid, what are the characteristics about it. So here this one is saturated with hydrogens, no carbon double bonds. Okay, here we have 1, 2, 3 carbon-carbon double bonds, so it's polyunsaturated, but they're all cis, where the hydrogens are on the same side. Now, this isn't drawn with the bends, but we know that there would be a bend here, there would be a bend here, and there would be a bend here. Okay, this one is monounsaturated. Okay, again, it's cis, so there would be a bend here if it was drawn accurately, but this is just drawn straight, so you need to know there would be a bend. Okay, here's just a couple of examples. Sunflower oil, 2/3 of the fatty acids are polyunsaturated, the rest are monounsaturated, so no saturated fats. Butter, on the other hand, 2/3 of the fatty acids are saturated, most of the rest is monounsaturated, so it's going to be solid at room temperature. Okay, so triglycerides are going to be for long-term energy storage. Remember, short-term was glucose, medium-term is going to be glycogen or starch. Triglycerides are used for long-term storage. Okay, these are going to be our fats and they're stored in adipose tissue. Okay, they're very chemically stable. They don't mix with water because they're nonpolar. But here's a massive benefit. They release twice as much energy per gram in cell respiration as carbohydrates. Okay, so enough energy can be stored in half the body mass. This is especially important for animals that move and especially for birds and bats that fly. If all of that energy was stored as carbohydrates instead of lipids, they would be twice as heavy and may not be able to fly. Or if you're trying to run away from a predator, right, you may not be able to run fast enough or run at all if you weigh twice the weight. And so lipids are going to be a really important form of energy storage that can be much, much more efficient in terms of body mass. Okay, lipids are also poor conductors of heat, so they can be used as a thermal insulator in animals. That's going to be especially important for marine animals. They're also liquid at body temperature. They can act as a shock absorber, for example, around the kidneys. Okay, there's lipids around the kidneys that if you were to get injured or struck in the kidneys near the back, that would act to protect them. Okay, so here's just an example of a marine mammal that would have a thick layer of blubber to act as a thermal insulator, okay, or animals like penguins that live in very, very cold locations. Okay, we have already covered quite a bit about phospholipids. Just remember they spontaneously form bilayers. They're amphipathic with a hydrophilic phosphate head and two hydrophobic hydrocarbon tails. Okay, here's a little bit more about steroids. Again, those four rings are that really critical shape. Note how few oxygens there are compared to each of these angles, which represent carbons. Definitely a lipid. Okay, we're going to skip through a couple of these that we would have done with our molecular modeling sets for now. And we're going to move on then to our next slideshow, which is all about proteins. Let's go. Proteins are made up of chains of amino acids. The amino acid order is coded for by DNA. The purpose of DNA, okay, is that every 3 bases code for one amino acid, and so it's the code for building proteins. Those amino acids then are going to eventually be joined together in a 3-dimensional shape, and we call them proteins. They're made by ribosomes, and the building of proteins is known as protein synthesis. Okay, you need to be able to draw and label a diagram of a generalized amino acid. Don't forget the alpha carbon is different from the carbon that makes up the carboxyl group. There should be 2. There's also going to be our R group, which is our variable group, the only thing that's different between the 20 amino acids. And then we have our amine group, which contains nitrogen. Okay, and this is going to have 20 different variations. You should be able to recognize and draw the amino acid. All of these different forms are the exact same thing. So whereas this is kind of an easy to learn cartoon version, okay, especially if you move on to more advanced biology or chemistry, okay, you're going to be using this notation. This means the exact same thing. But remember, anywhere there's an angle, there's a carbon. So right here is going to be our alpha carbon. Just like we see here, the alpha carbon is going to have an H, a hydrogen, and an R group. So that's what we're seeing here and here. Then it's going to have the amine group, the NH2. And then this is the carboxyl group. Here's that carbon double bonded to an oxygen and then also bonded to an oxygen and a hydrogen. And so that's what we see here in purple. So you need to be able to recognize this structure as well. Okay, so of the 20 amino acids, you can see they're going to have different chemical characteristics. Polar, nonpolar, positive, negative charges. Some will have sulfur, ability to form disulfide bridges. Okay, amino acids, when they link together through condensation reactions, 2 amino acids will form a dipeptide, okay, and 3 or more form then a polypeptide. Note that the way this happens, okay, is we have an OH from one amino acid that's going to bond with an H from this amine group. Okay, so then we have 2 H's and an O. That's where our water is going to form. And it's going to allow this nitrogen to then bond directly to this carbon. So the dipeptide bond is a carbon-nitrogen bond. You should be able to recognize and determine where peptide bonds form. An easy way to do that is just counting from one side. So here's our carboxyl group. Here's an alpha carbon. Okay, this is going to be an R group, and there's the H. Okay, here's part of an amine group, but we know that the H is gone along with the OH from this carboxyl group. So right here is a dipeptide. And if you were asked to, you would draw an arrow pointing to this bond right here, indicating it's a dipeptide bond. Okay, and there's other ones as well that you can identify. Okay, so here is just another way of taking a look at a dipeptide. All right, now of the 20 amino acids, 9 of them are considered essential. Essential amino acids cannot be synthesized by the animal and must be obtained directly from food. Non-essential amino acids, the other 11, can be made by transforming one amino acid into another. So it's going to be important that you are getting all 9 of your essential amino acids and just having a variety of amino acids in your diet. Now, if you do eat meat, you are probably going to get a balance of amino acids that's similar to what's needed in the human diet. Okay, however, if you are a vegetarian or vegan, especially a vegan, it's going to be really important that you have enough of each of those essential amino acids and that you make good choices with knowing about the different needs for these different amino acids. This is totally possible to do in a healthy way. In fact, there are traditional plant-based diets in successful civilizations that provide a balance. Here are some examples, but just a quick note that that's going to be important that you are educated and aware that you do need to have that variety of amino acids as a vegan. Okay, so we have an infinite variety of possible peptide chains because there are 20 different amino acids, okay, but they can be in any order and the polypeptide can be any length. And so you literally have an infinite different number of proteins that can be generated. Here are some examples. Beta endorphin is a natural painkiller secreted by the pituitary gland. It's made up of 31 amino acids. Insulin is a small protein that has 2 polypeptides. They're joined by 3 disulfide bridges. Alpha amylase is an enzyme that breaks down starches. It's found in your saliva. And a final example is titin, the largest polypeptide discovered, okay, made of 34,350 amino acids in humans. Very impressive. Now, a polypeptide can become denatured, and this is when its 3D shape changes and it is permanent. It cannot go back. Once a protein is denatured, the only way to get another protein is to make a new one from scratch. So how does denaturation happen? Well, one way it can happen is from heat. Heat causes vibrations that can break intermolecular bonds. An example of this is cooking an egg, where the albumin protein here is turning from clear to white as it's denatured. And as you know, you can't uncook an egg and go back to having egg whites. Okay, or clear, I should say, clear egg whites. Also, extremes of pH is another way that we can denature proteins, because remember, pH is based on hydrogen ions, which of course have a charge, they're protons, and so by changing the number of hydrogen ions in solution, that can mess up your ionic bonding because ionic bonds are based on charges. Okay, at this time, this ends the SL content for B1.2. We're going to continue on with HL content now. Okay, so once more, those 20 amino acids have different chemical properties. Okay, we're going to run through the 4 levels of protein structure. First up, the primary structure of a protein is simply the order of amino acids as coded for by DNA. Okay, but we can then move on to secondary structure, because at regular intervals along the polypeptide chain, there's going to be those C double bonded to O and NH groups that were left over from the amine and carboxyl groups after they were used to make a peptide bond during that condensation reaction. Now, both of these are polar, okay, and so therefore hydrogen bonds can form between these groups. And because of these weak hydrogen bonds, we end up with these substructures within the protein, these secondary structures. Okay, they can either be in the form of an alpha helix or a beta pleated sheet. These are common secondary structures found within proteins. Okay, but the tertiary structure, which is the full 3-dimensional shape of the protein, is based on interactions between those R groups. Remember, the R groups are those variable groups on the amino acid. Okay, and based on their chemical properties, it's going to form into the same 3D shape every single time. Okay, and this is going to be important especially for globular proteins like enzymes that need to have the exact same active site shape in order to react with its specific substrate. Okay, ionic bonds can form between positively and negatively charged R groups. Hydrogen bonds can form between polar R groups. Disulfide bonds happen between pairs of cysteines, which contain sulfur. And then hydrophobic interactions can happen with any of the nonpolar R groups. They tend to try to fold towards the center away from the water in the cytoplasm where it's being made by a ribosome. Okay, so we also might see questions on the test where you're asked about the amino acids that make up proteins embedded in a cell membrane. You should be able to identify that amino acids that are towards the outside liquid of the cell and the inside cytoplasm liquid of the cell are going to be hydrophilic versus the amino acids that make up this protein that are going to be towards the inside of the membrane where we have those nonpolar tails are going to be hydrophobic. Okay, also note that in a channel protein, we have the same thing where we have hydrophilic, hydrophobic here, but you should note that most of these molecules are going to be able to pass right through the membrane if they're nonpolar, and so these protein channels are going to be usually for polar molecules, and therefore we have polar amino acids that are going to line this channel as well. Okay, quaternary structure, our final level then is going to be when we have multiple polypeptides together. An example of this would be collagen, okay, which is the 3 polypeptides joined together that make that rope-like structure, provides that high tensile strength. We often talk about collagen in skin, keeping our skin firm and non-wrinkly, okay. Human insulin is another example. Now, these are both examples of non-conjugated proteins that are only made up of polypeptides. In contrast, we also can have conjugated proteins, okay, which can be made up of non-polypeptide subunits. This is an example of hemoglobin. It's made up of 4 polypeptides, so it has this quaternary structure, but it's also conjugated because it has these heme groups that are going to be carrying the oxygen as that hemoglobin is in a red blood cell traveling around the body. Okay, so here's a globular protein. Here's our fibrous protein. Let's compare the two together. Okay, whereas globular is rounded, our example here is insulin. Okay, that 3-dimensional shape allows it to perform a specific function. Fibrous is going to have, again, this collagen with that high tensile strength. Okay, here's a summary of those 4 levels of protein structure. Okay, moving on then to B2.1, SL students will need to rejoin. Cell membranes, we keep talking about them. That must mean that it's important. You should know all about the structure of those phospholipid bilayers. Okay, let's talk a little bit then about transport. So a bilayer, remember, has that nonpolar core and or hydrophobic core, right, from those tails. And so we're going to see that polar molecules like glucose, indicated here in blue, are going to have low permeability because they're polar. They're not going to be able to travel through this nonpolar layer. These chloride ions in green, also ions have a charge, so they're not going to be able to travel through this nonpolar region. Oxygen, on the other hand, is small and nonpolar. So oxygen, as well as carbon dioxide, are going to freely pass through the cell membrane because they're both small and nonpolar. Now, let's talk about our 4 types of membrane transport. First up is diffusion or simple diffusion. This is when particles move from high to low concentration down the gradient directly through the cell membrane. This is a passive process. Okay, particles are in constant movement. That's just physics. Okay, and so because there's more of them on one side than the other, as that movement occurs, more of them are going to go this way until we reach equilibrium, and we have the same concentration on either side of that membrane. We're still going to see movement back and forth, but of approximately the same number of particles or solutes. Okay, We also have these integral proteins, which are going to allow for what we are going to call facilitated diffusion. Similar to simple diffusion, it's still molecules moving from high to low concentration, but this time this is going to be for things that are hydrophilic, okay, that can't directly pass through the bilayer, and so they're going to need to move through these specific channel proteins, which are forms of integral proteins that span the entire membrane. Okay, osmosis, on the other hand, is that net movement of water. Now, this water is going to go from low to high solute concentration because it's trying to bring those two concentrations to equilibrium. Okay, now water is polar. Okay, it can pass at a very slow rate directly through the membrane just because it's small. It can kind of squeeze through. But if we want to have large amounts of water move quickly, we're going to need to send it through aquaporin proteins, which allow for fast movement of water. Okay, so here's that facilitated diffusion I mentioned earlier, where we have a channel protein. Again, molecules are going to move from high to low solute concentration along the gradient. So then our fourth type is the type that is not along the gradient. Active transport moves through protein pumps. We call them pumps when it's active transport because we're using energy to pump molecules against the gradient. Here we're going to have solutes moving from low to high concentration. So we're going to increase the concentration here. That's going to require ATP energy. Okay, so here's a nice summary of our three types of passive transport versus active transport. Okay, this is very important that you know these four types of transport because they are going to show up throughout the rest of the course, especially when we do a lot of our anatomy and physiology. We'll be talking about different examples of these types of transport in the human body with our body systems. Okay, quick note on glycoproteins versus glycolipids. Glyco, of course, refers to the carbohydrate. So here we have a carbohydrate attached to a protein embedded in a cell membrane. Okay, and here we have a carbohydrate attached to this phospholipid. Glycoproteins are there for cell-to-cell recognition. Glycolipids help the immune system distinguish between self and non-self cells. Okay, this is a drawing skill. You need to be able to draw a phospholipid bilayer, and you need to be able to indicate each of these different parts of that. Make sure that you include the cholesterol and that you have it aligned with the phospholipid bilayer. Okay, this takes us to the end of the SL content. So if you are an SL student, you're going to want to skip ahead to B2.2. HL students, let's keep going. So let's use what we know about saturated versus unsaturated fatty acids and apply it now to what we know about cell membranes. Now, if the hydrocarbon tails are saturated fatty acids, they're going to be straight, meaning that we're going to really pack together those phospholipids tightly. That's going to mean the cell membrane is going to be very viscous, it's going to be very strong, it's going to have higher melting points. Okay, in contrast, if we have several unsaturated fatty acids within the membrane, okay, the membrane is going to be much more fluid because they don't pack as tightly because of all of these bends. It's going to be more flexible at temperatures experienced by a cell. Low temperatures, it allows for more flexibility. Okay, and one way we see this is if we look at fish, for example, okay, that are adapted to live in very, very cold places. This is an Antarctic fish. Its membranes around its cells have been found to have a higher percentage of unsaturated fatty acids compared to fish from warmer waters because we don't want them to freeze, right? We want them to stay fluid even at cold temperatures. Okay, so let's talk about cholesterol. I like to think of cholesterol as a guy walking into a room in a super fuzzy sweater, and everybody in the room that's packed with people are wearing like sticky Velcro suits, bodysuits. Okay, what's going to happen? Well, all the people near that cholesterol, right, with their Velcro suits are going to stick to his super fuzzy sweater. Cholesterol works the same way where it reduces membrane fluidity. Think of these little phospholipids that surround it kind of sticking to it like that Velcro. These are all Velcro suits sticking to that fuzzy sweater. Okay, but cholesterol also then has a secondary role of when we get really cold temperatures, okay, it's going to help prevent stiffening and that fracturing at very low temperatures. And so it has that dual role of decreasing membrane fluidity, but also preventing that crystallization, that fracturing at low temperatures. All right, so the classic here, proteins being made in the RER, we're going to see that that membrane can bud off because this membrane here is the exact same as this membrane, is the exact same as this membrane. Because these are all made up of the same phospholipid bilayer, okay, we can have vesicles of that membrane full of whatever's in them budding off from some of these organelles. Then those vesicles can fuse with other organelles that are made up of that same phospholipid bilayer. Okay, in this case, we have proteins made by these ribosomes studding our rough RER, okay, budding off as a vesicle full of those proteins, headed over to the Golgi apparatus, which remember is like our UPS. It's going to modify and sort and send off our proteins. And so that vesicle is going to fuse with the Golgi, and then it's going to bud and fuse and bud and fuse and bud and fuse, moving all the way through the Golgi before it eventually buds off. And that vesicle can then fuse with the plasma membrane of this cell and therefore secrete whatever the protein is that was being made. Maybe it's a digestive enzyme, maybe it's a hormone, okay, but whatever it is, it's that cell's job to secrete that particular protein. Okay, another form of cell transport is endocytosis. Okay, this is where, for example, possibly a food particle, okay, is going to move towards this plasma membrane. The plasma membrane is going to engulf it. Okay, it's going to form a vesicle and then be able to be brought into the cell. Okay, the classic example of this is the paramecium taking in large food particles this way. Okay, in the placenta, proteins from the mother's blood, including antibodies, are absorbed into the fetus this way. Okay, and then white blood cells are going to be engulfing pathogens in this manner, which then those vesicles containing pathogens can fuse with lysosomes containing digestive enzymes, and those pathogens can get broken down. Now, this is endocytosis. Endo is a prefix meaning in. So the opposite also can exist. Exocytosis is where we go out, we exit. So here we have perhaps some of those digestive enzymes I mentioned earlier that the cell was producing, okay, and those can be expelled from the cell through endocytosis. The other example of that is then our paramecium friend again with those contractile vacuoles, okay, having large amounts of water be able to move out of the cell quickly. We also have gated ion channels, and we'll talk more about these when we do the nervous system, okay, but how an action potential is sent down a neuron, okay, is we're going to see a change in voltage, which is basically a change in these ions. Because ions have a positive charge, as we change the number of ions, we're changing that voltage. And a trigger of the change in voltage can open up this voltage-gated channel and allow for a flood of ions to move through it. Okay, we can also have these neurotransmitter-gated channels where here we have some neurotransmitters that can bind to this protein, which allows for that channel to open up. And we're going to see that in the postsynaptic neuron, that is how we're going to re-trigger the action potential and pass it from the presynaptic neuron to the postsynaptic neuron, from an electrical signal to a chemical signal, back to an electrical signal. Okay, now our classic example of active transport is our sodium-potassium pump. You are going to want to be very familiar with this. Okay, in the sodium-potassium pump, we use ATP energy because we're pumping an active transport against the gradient. We're going to pump 3 sodium ions out for every 2 potassium ions that come in. This 3-to-2 ratio is going to be important in establishing our resting potential of our neuron, which we'll learn more about later. Okay, and the way that we're going to remember which ions go in which direction is the sodium-potassium pump wants to make a salty banana. What do I mean by that? Salt, you may know, is NaCl, that's sodium. And so the sodium wants to go on the outside of the banana, so it's salty. And you may know that bananas are full of potassium. So the potassium is going on the inside, that's going to make the banana. So the sodium-potassium pump is trying to make a salty banana. Another form of transport, okay, this is going to be glucose and sodium co-transported into this cell, but this is driven by a sodium-potassium pump. The sodium-potassium pump is going to use ATP energy to pump sodium, okay, out of this cell, and so now we have low amounts of sodium, Sodium is now going to be able to facilitate a diffusion passively into the cell because we have low concentrations here because it's been pumped out, but it's going to co-transport with glucose so that glucose also enters the cell with the sodium. Okay, lastly, adhesion of cells to form tissues. All of our cells don't just like run around raucously. Instead, they form tissues, and that's because their cells are going to be adhering to each other. So here's an example of cell adhesion molecules, or CAMs. They're located and embedded in the plasma membranes, and they can join together. They're typically proteins, and they allow for tissues to form instead of just having individual cells that aren't connected. All right. Moving on to slideshow number 4, B2.2 Organelles and Compartmentalization. Time for SL students to rejoin. Okay, you need to know that organelles are discrete structures in cells. You also need to know how many membranes, 0, 1, or 2, each organelle has. Okay, you should know that cell walls are not considered organelles because technically they're extracellular. The cell ends at the plasma membrane. The plasma membrane, okay, is what determines what goes in or out of the cell, and the cell wall is outside of the plasma membrane, so it's technically extracellular on a plant or a fungi, which also has a cell wall. Okay, the cytoskeleton, which is within the cell, is not a discrete structure, so it's not technically an organelle, and nor is the cytoplasm, the liquid that makes up the inside of the cell. Okay, so this is just a little preview for protein synthesis. Okay, proteins are made by our DNA serving as the master code, okay, forming copies through transcription called mRNA, which then leave the nucleus, and ribosomes then turn that DNA code into a sequence of amino acids, which folds then into a protein. Now, one advantage of having a nucleus indicated here in dark blue is that we can modify that mRNA before it exits the nucleus to become translated into proteins. In prokaryotes, this isn't possible because prokaryotes don't have a nucleus, and so often you will see transcription occurring, and even as the transcription is still occurring, we already have a ribosome jumped on to start translating and making a protein. Okay, other advantages of compartmentalization. We can take enzymes and substrates for a particular process and have them become much more concentrated. So for example, enzymes that are needed to do the Calvin cycle are going to be concentrated in the stroma, the liquid inside of a chloroplast. Rather than floating all around about the cytoplasm, it would be much more inefficient for those enzyme substrate reactions to occur. Also, substances that could damage the cell, okay, can be kept inside the membrane of an organelle. For example, a lysosome is full of digestive enzymes. If those digestive enzymes were just floating around the cell, they'd start digesting parts of your self cells, which would be bad news. And so we contain it as its own organelle. We can also have specific pH conditions maintained for particular processes. For example, back to our favorite example, that paramecium, when it feeds by endocytosis, okay, that food is ingested into a vacuole, and the pH in the vacuole then drops below pH 2, okay, for optimum digestive enzyme conditions, and then it can return to pH 7 for depending on what the enzymes need at any particular time. Okay, that's the end of SL content for this 4th slideshow. There will be still one more, so skip ahead. HL students, a few adaptations of the mitochondrion for production of ATP by aerobic cellular respiration. Note that there are 2 membranes of a mitochondria. The inner membrane has these deep infoldings called cristae. The purpose of those infoldings is to increase the surface area of the inner membrane because we're going to see that the electron transport chain is going to occur along the membrane, okay, which is going to be important that we have as much membrane as possible so we can maximize our amount of the electron transport chain. Here you can see all those infoldings in an actual micrograph called cristae. Okay, so they increase the surface area. We kind of already covered that. Let's see. Now, number 2, there's only a small amount of space between the inner and outer membrane. Okay, when we look at aerobic cellular respiration, we're going to see that a proton gradient gets built up. And the smaller the space, the faster the gradient can build up. And so that's an adaptation to be able to build up the concentration gradient more quickly. Okay, and then like I mentioned before, Okay, the fluid of this matrix, this inner part of the mitochondria, is going to have enzymes that are needed for the Krebs cycle, and those enzymes are going to be able to be concentrated right here in the matrix of the mitochondria rather than throughout the whole cytoplasm of the cell. Okay, adaptations of the chloroplast, we're going to have the same exact 3 adaptations, but with slightly different structures. Note that chloroplasts also are double membraned, but their inner membrane doesn't have infoldings. Instead, inside of the chloroplast are these stacks of thylakoids that are made of these little membrane surrounded pouches, and they're small, so you can kind of already guess that might have something to do with the small space of the mitochondrial inner membrane space, and you would be correct. So, first up, you can see here these thylakoids have a very large surface area. This is where our photosystems are going to be located, where light is absorbed for photosynthesis, okay, and electron transport chains are going to be. And so the more membrane we have, right, the more efficient our light-dependent reactions can be. Okay, and like we said, a proton gradient is going to need to build up, and so the smaller that thylakoid space, the faster that gradient can build up. Okay, and then just like the mitochondrial matrix, this liquid part of the chloroplast is called the stroma, and the same thing where we have a buildup of enzymes, okay, in this case to be able to drive the Calvin cycle. Okay, the nucleus has a double membrane. You can see here that the membrane is going to fold over at the location of nuclear pores. Those nuclear pores are going to be important because we know that that mRNA is going to have to exit the nucleus, as well as ribosomes when they're created. Okay, but also proteins are going to need to enter. The proteins that are needed for DNA replication, transcription, okay, as well as proteins like histone proteins for packaging up of that DNA really tight and being able to control gene expression. So we're going to need to have these pores for all of those things to move through. A second advantage, though, of having 2 membranes is that when we do mitosis, our nuclear membrane or envelope is going to break down, which is going to allow for the separation of sister chromatids, okay, and when we're replicating a cell in mitosis. Because there are 2 membranes, that's going to allow for the double membrane to easily break down into vesicles. Okay, and then after those chromosomes are separated, those vesicles can then easily reform that double membrane and reestablish the nucleus. Okay, remember that free ribosomes that are just floating around the cell are generally going to be making proteins for use inside the cell. Okay, versus proteins that are made by the RER are generally going to be for excretion from the cell or potentially transport within the cell. The Golgi apparatus, our UPS. Okay, remember proteins are going to arrive in vesicles from the RER. Okay, those vesicles then are going to fuse with the cis face of the Golgi, and they're going to bud, fuse, bud, fuse, bud, fuse all the way through. Okay, as they go, enzymes that are in the cisternae just means these, how would you describe these membrane sacs that are going to be able to modify and change that polypeptide. They might add a carbohydrate to make a glycoprotein. Okay, and as they go through all this processing, they then eventually, those vesicles can be secreted through exocytosis outside of the cell, and that product can then do whatever it needs to do. Okay, then lastly, first structure of vesicles is clathrin is a 3-legged protein that helps for easy formation, fusion, budding of those vesicles. They form a lattice of pentagons or hexagons, okay, and it helps for the plasma membrane to become indented and eventually to form a vesicle that has this clathrin cage around it. All right, folks, last one. SL students, welcome back. Cell specialization. Okay, we all started as a single fertilized egg, and yet humans have 220 distinctively different cell types. So how is it that one cell becomes 220 different cells that are specialized for a particular job? These are just some examples of different jobs. You can see how different these different cells are. Okay, how does it know? Well, first up, that cell differentiation happens, okay, because of gradients of signaling chemicals. Those gradients of chemicals are going to indicate to those replicating cells what type of cell they should be differentiating into. Now remember, they're not going to lose any of the DNA. Every cell in your body has your complete set of DNA, but only certain parts of that DNA get expressed, meaning they get turned into proteins. So those gradients of signaling chemicals are going to indicate what type of cell that cell should become. Okay, stem cells are undifferentiated cells that have not yet fully specialized. Up until the 8-cell stage, those cells can be separated and can give rise to an entire organism. So stem cells divide endlessly with no limit to the number of times it can split. Okay, and they eventually begin to differentiate along different pathways. Okay, we do have some stem cells remaining in the adult body. For example, bone marrow, where we're continually making red blood cells, hair follicles, where we're continually growing hair. So that precise location of stem cells within a tissue is called the stem cell niche. Okay, and it allows for conditions where the stem cells can be maintained over long periods of time, and then when needed, they proliferate rapidly and differentiate. Okay, now stem cells that are within skeletal muscle remain inactive unless there's a muscle injury, at which point they become they start proliferating and differentiating. Bone marrow and hair follicles, on the other hand, are continuously producing those particular types of cells. Now, interestingly, there's a lot of research being done right now on induced pluripotent stem cells. Okay, so we're going to see that the more types of cells that a stem cell can become, the more potential uses it has in medicine and therapeutic uses. Okay, now before we jump into that, just to note, there are also possible non-therapeutic uses, interestingly, like for example, creating lab-grown meat. Would you eat it if it was made from stem cells? This could be the future. All right, let's look at our different stem cell types. First up, this word totus sounds like the word total. Totipotent stem cells are from those very early stage embryos, and just like the word total, they can differentiate into any cell type. Okay, totipotent stem cells could be used in the growth of whole replacement hearts, kidneys, or other organs. Pluripotent sounds like the word plural. This is going to be later in embryo development. They can differentiate into a range of cell types, but not every cell type. Could be useful in therapeutic treatments. Okay, but found in adults, we're only going to have multipotent. They're only going to be able to form a few different cell types, more restricted than pluripotent. So this is why we're studying ways that we can take adult multipotent cells and induce them to go back to become those same pluripotent qualities for additional therapeutic reasons. Okay, next up, let's look at some examples of cell size as an aspect of specialization. Okay, let's start by comparing sperm versus egg. Sperm are tiny and narrow compared to eggs, which are the largest cell in the human body. Now, this makes sense because the role of the egg, okay, is to store large quantities of food reserves. For that whole week, it's traveling down the oviduct, okay? It's self-sufficient until it is able to reach the uterus, okay, and form a placenta that can then get more nutrients from the mom. Sperm, on the other hand, their whole job is to swim and move towards that egg. And so they're going to be very, very simple, very narrow, okay, and very, very small because they're basically just the half DNA that's carried in the head, right? The mitochondria that's in the midpiece, and then the tail for locomotion. Okay, let's compare our two types of red and white blood cells. Red blood cells are going to be very small. They have this flattened shape with indentations. It's going to help red blood cells make their way easily through even the smallest, narrowest capillaries. White blood cells are slightly larger. These are going to be used in our immune response. We're going to talk more about those when we get to the immune system and some different types. Okay, neurons are cells that have huge variation. Some of them are super tiny. Neurons in the brain can be as small as 4 micrometers in diameter. Okay, whereas some motor neurons can go all the way from the base of your spine down to the tip of your toe, a single cell that can be more than 1 meter long. So incredible variation. Striated muscle fibers are going to be larger than normal cells, which allow those fibers to exert greater force than smaller muscle cells. Okay, surface area to volume ratios must remain large. You need to have a large surface area for the amount of volume within a cell, which is why cells must be small. So just remember, small cells have a large surface area to volume ratio. Also, we need to make sure that we have a short path of diffusion, which is a second reason cells need to be small, okay, which allows, because diffusion is a slow process, this allows for nutrients to diffuse in time to not starve, and for waste products to diffuse out before the cell poisons itself. So we need small cells with a large surface area to volume ratio and a short path of diffusion. Okay, that will be the end of SL content for this video. We have a few more things to cover for HL students. Okay, so let's look at some specific adaptations. We've mentioned a few of these. So again, with red blood cells, to increase the surface area to volume ratio, we have that flattening with these indentations on both sides. Okay, this is going to give a smaller maximum distance from any part of the cytoplasm to the plasma membrane. Okay, other adaptations include microvilli. These microvilli serve to increase that surface area, okay, and they're going to be found in tissues specialized for absorption. Similarly, these infoldings called invaginations do the same thing of increasing the surface area. Okay, another example of that is the alveoli that are found in these what look like clusters of grapes, okay, at the end of the bronchioles in the lungs. Okay, by having this shape, we increase the surface area that allows for gas diffusion to occur between the alveoli and the surrounding network of capillaries. Okay, so here's a close-up of the alveolar space within just one of those little grape-like alveoli. Okay, there are two types of cells that line the alveoli. There's type 1 cells, which are adapted for gas exchange. They're going to be long and skinny with a short path of diffusion and a large surface area. Type 2 cells, though, are specialized to produce surfactant. Surfactant is what prevents the alveolus from collapsing in on itself when you exhale. Here's a picture of what they look like in real life. Our type 1 gas exchange pneumocytes and our type 2 surfactant producing pneumocytes. Okay, muscle tissue we're going to be spending more time talking about in the future, but it's contractile. Muscles contract. They're made up of cylindrical structures called myofibrils. Okay, and let's compare striated skeletal muscle to cardiac muscle. Now, both are made up of those contractile myofibrils. Skeletal muscle or striated muscle is attached to bones throughout the body, and it's used to move the body, and they're long cells. Cardiac muscle make up the wall of the heart. The heart's job is to pump blood throughout the body, and the cells are significantly shorter. Okay, we know that striated muscle is multinucleated, whereas cardiac muscle is just going to have one nucleus per cell. Striated muscle is unbranched. Cardiac muscle is branched, okay, which you can see here, which allows for electrical signals to be propagated more quickly, which allows for synchronization of muscle contraction in the heart. Okay, so whether or not a muscle fiber should be classified as a cell is debatable. Okay, they do have a plasma membrane that encloses them, but remember, they do have many nuclei and they're larger than most animal cells. Okay, adaptations of sperm and egg cells. These are our gametes, our sex cells. They each contain a haploid nucleus, and when they fuse together, we call that fertilization, and they form a zygote. Sperm are active movers, egg cells are moved passively and slowly. Egg cells have food reserves, sperm have little or no stored food. So let's break down some adaptations to wrap up this video. Okay, egg cells. It's going to be very important that only one sperm are able to enter the egg. If more than one sperm enters, it becomes inviable. And so there are going to be some mechanisms that only allow for one. First up, surrounding the egg cell is the zona pellucida, a layer of glycoproteins that sperm can penetrate, but after fertilization, it's chemically altered to prevent additional sperm from entering. Okay, we have binding proteins located in the plasma membrane that help the membrane fuse with the membrane of the sperm, allowing the nucleus to enter the egg. Now, surrounding, you can see here, these little orange circles are cortical granules. These are vesicles that contain enzymes. They're located near the plasma membrane. When the first sperm enters the egg, those cortical granules immediately release their enzymes into the zona pellucida, which prevents any additional sperm from being able to enter. It becomes impenetrable in order to make sure we don't have multiple sperm in the same egg. Okay, egg cells also have that large yolk, a large volume of cytoplasm inside the egg cell that contains stores of lipids and other foods, okay, to sustain the egg on its journey down the oviduct. They have mitochondria that produce ATP. They divide repeatedly to generate all the mitochondria in an adult's body. Those all came from your mom's egg, so all of your mitochondria are from your mom. So if your mom is still around, you should say, "Hey, Mom, thanks for my mitochondria. I really appreciate that. They can be the site of my aerobic cellular respiration." Okay, also centrioles that are going to be needed for mitosis in the egg. Okay, on the other hand, the sperm is much more simple, adapted for rapid swimming. It's got this tail, this long flagellum with microtubules. Okay, the midpiece has mitochondria to provide those large quantities of ATP needed to make the tail move. Okay, the head is streamlined in shape. Here's our tightly packed chromosomes. And then we have our acrosome. Okay, the acrosome is the sac of enzymes that's going to digest proteins in the zona pellucida so sperm can reach the membrane of the egg. There are also receptors for the binding proteins we mentioned earlier that are in the zona pellucida, and binding proteins themselves, which are revealed after exocytosis of the acrosome, binding to proteins in the plasma membrane of the egg, leads to fusion of the membranes and entry of the sperm nucleus to the egg. All right, that wraps up B1 and B2. Thanks for listening.